Hello, my name is Cynthia Webster. I'm from the University of Connecticut, and today I will be discussing challenges of genome annotation in plants and the development of our eukaryotic annotation tool, EASEL. Now, we define genome annotation as a process of correctly identifying protein coding features. And plant genomes are especially difficult to annotate because they often have larger genomes as a result of increased presence of transposable elements and polyploidy. And we wanted to benchmark these challenges, so we selected 5 plant genomes with variation in genome size and repeat content, including traditional models such as Arabidopsis and more complex models with higher fragmentation like Liriodendron. We then annotated all 5 species with common tools such as Maker and Breaker, using short and long RNA reads as well as de novo assembled proteins. And in this example, looking at model species populace, notice that the Breaker-Busco scores shown above are nearly identical, but mono to multi ratios are fairly different. This indicates that common metrics might detract from issues of fragmentation. Also notice a significant difference between Breaker and Maker despite the same input, as well as how additional structural repeat annotation and addition of long read data improve the genome annotation. As a result of Vidya's work, we found that commonly used pipelines are often time-consuming, challenging to run, and require significant downstream post-processing to generate annotations. So here we present EASEL, a eukaryotic genome annotation tool that leverages machine learning, RNA folding, and functional annotations to enhance gene prediction accuracy. The first step of EASEL is preparing a soft masked genome. Next, RNA evidence is trimmed and aligned to the masked genome. And a dual approach for transcript generation is utilized, and complete open reading frames are identified with homology to known proteins via eggnog. Non-redundant complete open reading frames are then aligned to the masked genome to produce our gene model to be trained by Augustus. Part 1 of machine learning is training Augustus with a subset of our gene model. And because we are predicting gene sets for Arabidopsis, we can compare our prediction to the reference, giving us an F1 metric at gene and transcript level. Notice as we incorporate protein and transcript evidence, we are left with higher quality predictions, which can then be combined into a single gene set. Part 2 of machine learning is start site prediction. We begin with a small sample of true start sites and ATGs that are not start sites and look up and downstream. Within our window, we can identify features such as Kozak consensus, GC content, and RNA folding structure. Using features and true labels, we use a random forest classifier to leverage creating uncorrelated models, which helps with the prediction of start sites. Refinement is still under development, but we plan to utilize the following tools to produce an official output. Overall, EASEL has made a significant progress in refining implicated gene structures and reducing runtimes. With that, I would like to thank everyone listed here. Please follow my Twitter and check out our GitLab for the project. Thank you very much.